Hello, my name is Henri Chung. I am a graduate student at Iowa State University studying bioinformatics and computational biology. And today, I will be presenting my project, which shows network-based analysis of microbial function reveals putative bacterial pathways. The underlying premise of this project is that identifying microbial function is essential to understanding the interactions between microbes and their host, their environment, and other microbes. Protein databases record some of the known functionality of microbial organisms, but for this information to be of use, there is a need to understand the interactions between microbial proteins, which we will refer to as pathways. To identify these pathways, we developed a novel network-based approach using phylogenetic profiles to determine functional relatedness. Below is a model depiction of the network, and in the following slides, I will go over the information used to construct the nodes and edges. Every node in our network is a collection of functionally similar bacterial proteins identified by HFSP, or homology-derived functional similarity of proteins. We start with an initial dataset of 15.6 million bacterial proteins retrieved from GenBank. After some initial preprocessing and filtering, we align these proteins based on their sequence identity and length. We then threshold the alignment scores and cluster similar proteins together, creating groups of functionally similar proteins, which we call fusion clusters. For this analysis, we take the largest 50,000 fusion clusters as nodes in our network. The edges connecting the nodes in our network are based off phylogenetic profiles, or a binary vector encoding for each fusion cluster representing the bacteria from which contributing proteins came from. Shown is an example fusion cluster and its corresponding phylogenetic profile, where each element in the profile represents a bacteria in our dataset. The values of the elements are either 1 or 0, indicating whether that bacteria contributes a protein to the given fusion cluster. In this case, our example fusion cluster is comprised of 3 proteins, each contributed by the light blue, yellow, and dark green bacteria respectively. We can construct phylogenetic profiles for each of the 50,000 fusion clusters in our dataset, and then we can calculate the Jacquard similarity between any 2 fusion clusters. For example, in the first 2 rows, we observe a partial overlap between contributing bacteria among the genomes, and this partial overlap can be quantified as a Jacquard similarity of 0.6. For rows 3 and 4, which are in complete agreement, their Jacquard similarity is 1. And for rows 5 and 6, which are in complete disagreement, their Jacquard similarity is 0. These values are used to calculate the edges in our network. Once we have constructed our network, our goal is to identify communities or groups of functionally related proteins which could represent possible bacterial pathways. We do this by first thresholding the edges in our network, removing extraneous edges between distantly related nodes. Here, we evaluate the unclustered network prior to community detection by determining if the edge weights connecting known bacterial pathways are significantly shorter than random. For this, we use KEGG, the Kyoto Encyclopedia of Genes and Genomes, which provides information on bacterial pathways defined by their contributing enzymes. We look at the edge weights connecting enzymatic proteins involved in the same pathways and determine whether the distance connecting those nodes was in fact shorter than an equally sized random group of enzymes. What our analysis showed was that our ability to detect these pathways was dependent on the number of bacteria in our dataset which contained that pathway. In the graph, each point represents a bacterial pathway. The y-axis indicates the significance of edge weights connecting that pathway, and the x-axis indicates the number of bacteria in our dataset in which the complete pathway can be found. As you can see on the graph, we observe a clear curve where the more conserved a pathway was, or the more bacteria which contained all of the pathway's enzymes, the more likely we were to determine its edge weights to be significant. Similarly, the less conserved a pathway was, the less likely we were to find it significant. This supports our underlying premise that phylogenetic profiles can be used to identify functional pathways. We then apply a community detection algorithm, in this case Louvain, a hierarchical clustering algorithm which identifies clusters by optimizing the modularity score of detected communities. We can then separate these communities and evaluate how well they reflect known bacterial pathways. We take our identified communities and determine if they were reflective of known pathways by calculating their protein-protein interaction scores. For this, we use String, a database of protein-protein interactions, and what we found was that the average combined interaction scores between proteins in our detected communities was significantly greater than that of random clusters. The average combined interaction score of proteins in our communities was 0.67, with half of the communities having an average interaction score greater than 0.72. This is significant because these clusters were identified entirely based on the phylogenetic profile-based edge weights, without any prior information for functional relatedness. Here, I would like to highlight an example community where our network has identified these 5 proteins as interacting. And while the String database contains interaction information for the first 4 proteins, all components of a Cas system, there is no reference for this 5th protein, a putative transposase interacting with this Cas complex. However, there is literature information documenting that transposons are known to interact with Cas systems. This suggests that our network is able to identify putative protein interactions which are not previously known, or at least in the String database. In summary, the goal of this project was to identify putative bacterial pathways using a network built with functionally similar proteins and phylogenetic profiles. We showed that our network is reflective of known bacterial pathways and interactions as defined by KEGG and String, and we present a novel way of identifying previously unknown protein interactions. This concludes my talk. I would like to thank the organizations and lab members who have supported me in this work. I invite anyone listening to come and discuss my project at my poster on July 11th, 12:30 to 2:30 p.m. in Ideation Hall, poster session A, poster number 4. If you would like a copy of these slides, please use the QR code shown on screen. Thank you.